not your usual host for this, but once in a while the science faculty let me come over and play in the sandbox over here. I'm Marion Brown. I head the Center for Sustainability, and I love to program science colloquium talks that find that intersection between sustainability and the natural sciences. And so this talk today does that. We're really pleased to welcome Jenny Bloodgood. She's a wildlife veterinarian in the New York State Wildlife Health Program and an assistant professor of practice in the College of Veterinary Medicine at Cornell. Prior to joining Cornell, Jenny was a research veterinarian at Dauphin Island Sea Lab, where she oversaw stranding response, necropsies, and disease research focused on marine mammals in Alabama, and offered educational opportunities to agency partners, volunteers, and students. She holds Bachelor of Science and Master of Science degrees in Wildlife Biology from Clemson University, a PhD in Integrative Conservation from the University of Georgia, and a Doctor of Veterinary Medicine from the University of Georgia with a focus on research and wildlife health. As both a wildlife veterinarian and biologist, she has broad interests in free-ranging wildlife health and disease. She has particular interest in pathology, infectious disease, and the interface of human and wildlife health. And she enjoys mentoring students and working in interdisciplinary teams to tackle real-world issues. Please help me welcome Jenny Bloodgood.
Thank you. Awesome. Thank you, Marion, for that introduction. So today I'm going to tell you a lot about what I do on a day-to-day basis, which when I'm not sitting at a computer, I do get to occasionally do some fun things out in the field, so those are the parts I'm going to share with you today. But I am a veterinarian with the Cornell Wildlife Health Lab, and the Wildlife Health Lab is a partnership with the New York State Department of Environmental Conservation. Together, we form the New York State Wildlife Health Program. So basically, the DEC does not have a veterinarian, so I sort of fill that role for them as a wildlife veterinarian. And our program does research and disease surveillance on free-ranging wildlife throughout the state of New York. So today I'm going to give a little bit of a background about the Wildlife Health Program and then jump into some sort of case examples of things that we work on a day-to-day basis. It might feel like a little bit of a whirlwind because I'm going to try and cover a lot of different topics, but bear with me on that. Hopefully they're interesting to you guys. So the goal of the New York State Wildlife Program is to really promote the health and sustainability of wildlife populations. And Marion mentioned, in my introduction that I love to work in interdisciplinary teams, and this is a great example of an interdisciplinary team. So we not only work with ecologists and veterinarians, but also with statisticians and biologists, and it takes interdisciplinary teams to tackle problems. I think having different views from different lenses is really important. The program was started in 2011, so we're coming up on 15 years old, and I started with the program just over a year ago, November of 2022. We have 9 program focus areas, and I'm not going to go through all of these, but I'm really going to highlight the surveillance and monitoring aspects as well as the preparedness and response. So surveillance and monitoring really focusing on infectious diseases and non-infectious diseases of wildlife, and then being prepared to respond in the event of an outbreak is really important. And then of course we do a lot of these things sort of, mesh together to accomplish these goals, like the communication. So public communication and outreach is a big component of what we do, as well as research and policy. So the big overarching topic here is One Health. It's always about One Health with me and our group. So One Health, if you're not familiar, is just the fact that animal health, human health, and environmental health are all inextricably linked. You can't just look at them individually. They all affect one another. And some of the examples of things that we do active surveillance for in our group to be prepared are things like avian influenza, rabies, chronic wasting disease, so lots of infectious diseases here, but also some non-disease things like human interactions with wildlife, and non-infectious disease like contaminants such as lead and PFAS. And the ones in red here are some of the ones that I'm going to highlight today. So this is just an example of our lab and partnership with DEC, some of the cases that we've looked at. This is just 2022. We had over 1,600 cases submitted, and the way that it usually works is a lot of these cases are deceased animals, so that the public or a veterinarian or a DEC biologist finds out in the wild, and they can be submitted through the DEC. And then through some criteria, we don't take everything, but we take a lot of things, they will be submitted to us. And by us, I mean we have 3 labs across the state, so one based at Cornell University, where I am, one in Del Mar, which is outside of Albany, and that's run by the DEC, and then one on Long Island, which is historically run by Cornell and is still, as of now, run by Cornell, but the pathologist actually migrated up to Ithaca and works with us directly now. But you can see here, most of our animals are birds that are submitted, and then followed by mammals, and then we do get a small percentage of herps, so reptiles and amphibians as well. Our monthly caseload, you can see 2022 is this yellow line, and it's interposed over our 5-year average, which is the dashed line, and then the standard deviation of our average. You can see 2022 was a little bit of an abnormal year. We had more cases than normal. Does anyone want to guess? Avian influenza. So we got lots and lots of birds and mammals actually submitted in 2022, leading that to be one of our higher years on record of submissions. And you can see from this chart the most common diagnostic categories. So these are causes of death that we've come up with in our lab. So once an animal comes to us, it gets a full necropsy, which is an animal autopsy to figure out why it died. Lots of those samples from those animals then get tested for infectious disease or non-infectious disease like anticoagulant rodenticides and things like that to try and figure out why they died. And these categories are the ones for 2022, and you can see that the most common, so we do have some that are, can't be figured out. We can't figure out the cause of death in every case. But of the cases we can figure out, the most common are trauma and then viral in 2022 because of avian influenza. And trauma includes things like being hit by a car, being electrocuted, a window strike in a bird species. And the interesting part of that to me is that that's really a category that we can help mitigate because there's things that we can do about that, as opposed to a lot of times like nutrition in a wild animal is something hard for us to work with, but we do our best. So to jump into a example of one of the things we work on, avian influenza, classic One Health issue here. So I love this diagram because it really shows how it's a One Health issue. It not only affects domestic animals like chickens, but also wild animals like whales, which is interesting to me, and humans, which is interesting to everybody, right? And on our webpage at the Cornell Wildlife Health Lab, you can find a lot of these really cool fact sheets. So I always like to share this, and I have a QR code later in the presentation that you can scan, or you can just go to our webpage and find fact sheets on a lot of different diseases, and we're constantly working to develop more. But they're basic, the basic information for the general public about what the disease is, how it's transmitted, if it can be treated, things like that. So avian influenza is caused by water birds. They are a natural reservoir, which means that they can carry the disease naturally. They can carry it symptom-free. So they may actively be shedding virus, but not appear sick at all. And that's generally how the virus spreads around the world. So waterfowl, shorebirds, they migrate, right? Long distances, and they can carry the virus with them during migration and spread it to a new place. The virus is shed in feces and saliva, and it gets into the environment in that way and can be ingested or inhaled by an unaffected individual who then becomes diseased. And the clinical signs range from inapparent in some birds to severe and can include death. So I am going to show some pictures throughout the presentation, a couple of videos coming up that can be a little disconcerting, just so that people are aware. But on the left here, you can see a Canada goose, and the point of this picture is to look at the eye. So it's cloudy, right? Can you tell kind of that bluish hue to it? That's a sign of corneal edema, and they've actually done experimental infections in some duck species and found this with avian influenza, and some researchers have been able to isolate viral particles from the eye. The next image, or it's a video, let's see if is another goose that is displaying some neurologic signs. So it's not able to really know where it is in its environment, and it's stumbling a little bit. And then we zoom in and can kind of see that it also has that opacity. And these are from the Swanson Wildlife Hospital, which is on Cornell's campus. They were nice enough to share these images. And then this bald eagle, might not work. Let's see. See if I can get it to work. Maybe it won't work. But the video shows the bald eagle has some head tremors going on, so it also has neurologic signs. So this is a breakdown of taxonomic orders of birds that were submitted to the Wildlife Health Lab, and a couple of mammals as well. This is 2022 to 2023. They're color-coded by their order, and you can see that a lot of water birds like ducks, geese, and swans, and shorebirds are sort of the most common species that we saw with avian influenza. But then there's this big group of raptor species. And bald eagles, there were 25 bald eagles that came in that had avian influenza. And this is actually a very low estimate of the total number, because I am only including animals here that were confirmed and sequenced as clade 2344 by the National Veterinary Services Laboratory. So avian influenza is a reportable disease, which means that if a lab tests and finds a positive sample, to really prove that, it has to go to the National Veterinary Services Laboratory in Ames, Iowa. And that's because it's reportable because of its effects in poultry. So highly pathogenic avian influenza, it's classified as high path or low path based on its effects in poultry. And high path means that it kills poultry. And so the federal government pays a lot of attention, because the poultry industry in the United States is huge, right? So bald eagles, we had a lot of cases, and there was a really interesting study, if you guys want to check it out of the University of Georgia with the Southeastern Cooperative Wildlife Disease Study. That's the one that I'm highlighting here, showing that highly pathogenic avian influenza infection was associated with decreased nesting success in bald eagles. So it's affecting them on a population level. And then I wanted to briefly talk about these mammals. So one interesting thing besides the raptors about this particular outbreak of avian influenza is its effects in mammals. So this has happened in previous outbreaks of avian influenza, but not to this extent. So we saw a lot of red foxes in New York. They were sort of the first species of mammal. And then we had 19 over the 2-year period. It started in kits, in the young animals, and we thought maybe it was, they're immunosuppressed or something like that. But then it started showing up in adults too. So unsure why foxes seem to be so affected, but possibly they're a peri-domestic species. They live around people. Maybe we see them more often and notice that they're affected, or maybe they really are more affected. They scavenge things. They'll eat dead birds. So that's a possibility as well. So in New York, this map shows the counties of New York, and the green are wild bird detections. The purple is captive and wild, and then yellow is captive only. And I put little stars in all the counties where mammals were detected. So it's pretty widespread across the state. The first detection was in a bird, a sanderling, a little shorebird on Long Island. And then our first mammal was a red fox, actually in Tompkins County. And then just to zoom out a little bit, this is the whole United States, the USDA. So as I mentioned, samples have to be sent to the NVSL for confirmation testing. And these are cases across the US that have been confirmed by MVSL. So basically, you can see every state is affected. The only place, United States state, that has not been affected is Hawaii. And this is just a 30-day snapshot. So as of February 20th this year, the last 30 days, this many cases have been seen in, these are wild birds. So I'm not even talking about domestic chickens and turkeys, right? In the outbreak in the last 2.5 years, there have been over 89 million affected poultry across the US. And then this is mammals. So this one's just extra interesting to me because it's mammals. But you can see the legend here on the right, the different species, and that's sort of still growing. It's simmered down a little bit, this outbreak has. But you can see here, the most interesting ones to me are the marine mammals because of my background, and I have an interest in marine species. I was actually still working at Dauphin Island Sea Lab when this dolphin, it was a bottlenose dolphin, died in Florida. It had clinical signs. It was swimming erratically and died during, before they could rescue the animal. And it turned out to have avian influenza. And then not much later, in Maine, there was a huge outbreak in harbor and gray seals, where many animals died, and they actually termed it, so the National Oceanic and Atmospheric Administration oversees cetacean and pinniped responses in the United States, and they termed this an unusual mortality event, which basically means more animals than usual. There's certain criteria, but are stranding and dying, and they put that allocates a lot of funding to figure out why. So there was funding put into this, and they tested a lot of the animals, and they were positive for avian influenza. And then the same thing out on the West Coast, harbor seals. And this issue is not relegated, this map only shows the US, but it's a worldwide issue, especially in marine mammals in South America. There have been thousands and thousands of elephant seals have died from avian influenza. So it's definitely having population level impacts still. So I'm going to talk a little bit about marine mammal strandings because it's kind of my baby, but marine mammals are a sentinel species, so we care about them because, not only because they're awesome and really cool, charismatic megafauna, but because they're sentinels of the ocean around them. So they're apex predators, they, top of the food chain, they live a long time. That means that they bioaccumulate and biomagnify things in the water, so they're really good sentinels of what's going on around them, of ocean health as a whole. And if we can identify threats to them, then we can identify threats to the ecosystem as a whole and use that to, design policies and management to hopefully mitigate those threats. And I always put up, when I talk about mammals, I always put up this little box with the stranding hotline. So a stranded marine mammal is any mammal that's out of its normal habitat, it's on shore when it's not supposed to be, or it's sick or entangled in some sort of gear or marine debris in the water. And if you see a stranded marine mammal, you should call this hotline because that is what connects you to the responders who would go and rescue that animal. And a side note, you should never push an animal back into the water if it is stranded. Everybody, you see these videos of people pushing an animal back into the water and it's like, yay, it swam off. Actually, about 95% or more of the time, that animal will re-strand because there's a reason that it's stranded in the first place, and you're potentially just prolonging medical care for that animal. So if you see an animal on the beach that's not supposed to be, like a dolphin, you should call this hotline and get medical care. So this is a paper that came out at the end of last year on some work I did on Dauphin Island about causes of death in bottlenose dolphins following the Deepwater Horizon oil spill. So the oil spill was in 2010. We saw effects of the oil spill. It was termed an unusual mortality event as well until 2014. And so we looked at causes of death post the UME after the oil spill. And the three most common causes of death were infectious, human interaction, and prolonged freshwater exposure. And I love this picture because it really illustrates what a one health thing dolphins and marine mammals are, because these are beach houses where a lot of people vacation, and that's a dead dolphin. So people are, in the same areas. They're often the first people to notice a dead dolphin. So it's, things that affect them affect us as well. And since we already talked about some infectious things, I'm going to just highlight human interaction and prolonged freshwater exposure next. So these are a couple of pictures of animals that I got to work on while I was at the Sea Lab. A good example of human interaction is fishing gear and marine debris in the oceans. And animals that live in the ocean can often ingest this marine debris, leading sometimes to their death. Sometimes it's incidental that you find a piece of plastic in a stomach or similar. But in this case, the animal on the left is a sperm whale, and that gear was huge. The scale is every black and white line together is a centimeter. So that gear is about 2 feet long, just to give you some perspective. And it was blocking the animal's stomach. And then on the right, this is from a manatee, so not a dolphin or whale, but I got to work on manatees as well. And that's a plastic grocery bag that was blocking the animal's esophagus. So marine debris is a big issue, and we should all do our part to try and prevent trash from ending up in our oceans. Another example of human interaction is entanglement. So dolphins will sometimes learn that they can get an easy meal by predating out of fisheries nets, and sometimes that leads to them getting stuck and drowning in a fisheries net. So this is an animal that did die in a fisheries net, and these lines, I'm not sure if you can see them very well. Is there a way to turn the front light down? I wonder if the pictures might show up better, but okay, all right, great. So there's some lines on the rostrum, the front of the mouth there, that are indicative of a net, impressions on that animal's skin. And then finally, another example of human interaction is actually being hit by a boat. So manatees are the classic example of this, and this is a study by some colleagues out of Florida that they showed that almost 100% of manatees have been hit by a boat at some point in their life by the time they're an adult. 1 in 4 adults had been hit more than 10 times by a boat, and 5 of, I think they looked at about 2,000 carcasses, 5 of those animals had evidence of more than 40 boat strikes. And the way that they know this drawing here matches the drawing up here, and I'm not sure if you can see, but there are marks on the animal up here that they've taken like a grease pen to highlight scars, and then they translate it onto a piece of paper and draw, and the different colors represent different boat strikes. And we know this because if a boat hits an animal, those parallel lines are propeller marks, sometimes accompanied with a straight line. So here, a straight line with some parallel, which is the bottom of the boat or the skeg in the back. And we can tell that the parallel lines going different directions are different boat strikes. So that was a little sad. So what can we do about this? And these are just some examples. So on the left, we worked with NOAA Fisheries to develop things like this that help people know, you need to respect marine mammals. So working with the public and public education is really important. Don't throw your trash in the water. You should stay 150 feet away from a marine mammal and give it a respectful distance. From whales, you're actually supposed to stay 300 feet away and observe from a respectful distance. We also created these signs in the middle for manatees because a lot of people in our waters in Alabama, the water's not like in Florida. It's cloudier, murkier, and so you can't see manatees that well, but they're there. And so a lot of people, we gave out these signs for free for people to put on their docks and say, I have manatees here, please go slow. And then I grabbed this one off the internet, but making sure, to throw your trash away, pass up plastic when possible so it stays out of the oceans. So Prolonged freshwater exposure was one of the other leading causes of death in our animals. And just to talk about that a little bit, you can see the photos on the bottom here. These are examples of dolphins that were exposed to freshwater for a long period of time. So dolphins, they're oceanic, right, marine species. Some of them are adapted to estuarine habitats and deal with freshwater, but when they're in freshwater for a long time, they don't seem to notice, and they can develop skin lesions like this. And these skin lesions can lead to secondary infections, they can get osmotic imbalances, and ultimately they can get kidney disease, cerebral edema, and die from freshwater exposure. So our group, being in Alabama with Mobile Bay, which is a highly dynamic estuary, we were really interested in the effects of freshwater on marine animals, on dolphins particularly. So you can see the blue dashed line here is discharge, so freshwater discharge into Mobile Bay. The green line is the associated salinity, so you can see when the discharge goes up, the salinity goes down. And then the bars across the bottom, the dark ones are strandings that had signs of freshwater exposure, and the white ones are ones that did not. So in general, animals with these skin lesions were stranding after periods of low salinity. And the next step in this project is to associate sort of the magnitude in the change of salinity and the duration of how long it stays low to dolphin deaths, to try and better understand, what can they tolerate versus not. Because we have seen animals recover. So we've seen animals, we do a lot of live dolphin work too, but we've seen animals with skin lesions multiple times in the wild. Their dorsal fins are uniquely identifiable, so you can tell the same dolphin. And we've seen them recover, have skin lesions, and later not. So we're interested in learning more about what they can tolerate. And a PhD student down there that I co-advise is studying the effects of freshwater on ecosystem health. So this is her image, which I love how she put this together, because you've got, humans, the animals in the water, and then all the other things, the environmental things in the water. And freshwater, you think about rainfall and snow melt coming into the bay, but you don't really think it's not just fresh water. It brings a lot of stuff with it, right? So it can convey things, she's got contaminants like mercury, agricultural contaminants, pesticides, you've got pathogens that it carries. So we had 2 dolphins that died from a pig-borne pathogen, and we were able to do genetic analysis of that and trace it back. It was most related to pigs, and probably came from feral hogs living around Mobile Bay. Freshwater probably came in a large influx and scooped some poop into the bay, and 2 dolphins died as a result. So, that just shows how interrelated everything is. So these are some of she's looking at a bunch of different aspects in both the water and dolphins. All right, I'm going to switch gears again, and now we're going to talk about bald eagles again, and lead poisoning. So an example of a non-infectious disease, right? So lead ammunition is used by the vast majority of hunters who hunt white-tailed deer, and these bullets are designed to fragment on impact. So it doesn't matter if you hit the animal, on target, the lead will spread up to 30 centimeters from that wound. And when you field dress an animal, which basically means you take its gut contents out in the field so that you don't have to carry a very heavy thing back, some of those lead fragments can end up in those gut contents and are left behind in the environment. And people call it an attractive nuisance. It attracts wildlife, it's a free meal out there. And a lot of times bald eagles are scavengers, so they see a free meal and they're gonna swoop down and eat that, and there's lead fragments in there. So this is an example of a deer that was shot, and all these sort of brighter white tiny little dots throughout the abdomen are lead shots, so thorax and abdomen there. And this is an example, so bald eagles will eat that, and not sure if you can tell, but there's the same sort of bright white fragments in the animal's GI tract this time. So this shows that the bald eagle ate that gut pile and ate the lead, and that's how bald eagles are more affected. They're not affected because they get shot by lead. They're affected because they eat lead. And lead as small as a grain of rice can kill a bald eagle. So they have a gizzard. They grind their food items, and that lead gets ground up, and then their acidic stomach acid sort of breaks it down, and it can kill them with a very small amount of lead. So we worked on a study across 7 states, and 78% of eagles were exposed to lead that we looked at in that study. 37% of those had toxic levels of lead, and they were able to translate that mathematically to a 5% decline in population growth. So my colleague Kristen Schuler says it very well. I think bald eagles, we always think they're like the success story, right, of the Endangered Species Act. They're recovering, they're doing great. That is true, they're recovering, but it's like driving forward with your foot on the brake. So they're not recovering as well as they could be. And this QR code here is a link to that study if anybody's interested in that. And back to deer. I told you we were going to jump around a little bit. So chronic wasting disease, I wanted to touch on. It's a transmissible spongiform encephalopathy, which is a mouthful, but it's a prion disease. So a prion is an infectious protein, and if y'all are probably too young to remember mad cow disease, but look it up. It's in the same class. It's a prion, and it's fatal to the animal. So the prion, as an infectious protein, it causes normal proteins in the body to mutate into the infectious variety, and it's called spongiform because basically microscopic holes start to develop in the brain matter, and they start having clinical signs. They can harbor the disease for years, which is unfortunate because they can actively shed prions in their poop and pee before they are sick looking. And then once they are affected, though, they generally die pretty quickly from the spongiform encephalopathy. It affects mainly cervid species, and around our area, it's white-tailed deer and moose primarily. And they shed the prions, the sick ones do. The prions can remain infectious in the soil for quite a long time, so it's very hard once it's in an area, very, very hard to eradicate it. Those infected animals are more likely to be killed by a predator or a hunter or hit by car. And we worry, people hunt white-tailed deer all the time and eat them. So mad cow disease was passed from eating affected cow meat. And could CWD be the same? It's a prion as well. So far, there's no evidence of that, but like I said, it can be harbored for like 15 years up to before showing clinical signs in an animal. So we think there's a pretty solid species barrier and people aren't likely to be affected, but it's always possible. So even the CDC recommends that you don't eat an animal that you know is positive for chronic wasting disease. This is the spread of the disease across the country and Canada over the last 20 plus years, and you can just see how quickly it started spreading. And New York, you'll notice, is red, somewhere over here, there we go. But we are the only state to have actually successfully eradicated CWD. So we had it at one point, so we have this blemish on our record, but we have eradicated CWD, and it does not currently exist in the state, and we are pretty confident about that because we do active surveillance for it all the time in hunter-harvested samples. We collect lymph nodes from the animal, which is the sort of gold standard for diagnostics for the disease, and we test them and have not found CWD in the last over 10 years. But this map is out of date. Unfortunately, in 2023, 3 more states were added. So it continues to spread and be a worry. So our group, to address this challenge, came up with this super cool thing. So this is straight out of our lab. I didn't participate in it, but I like to talk about it because it's really neat. The SOP for CWD, the Surveillance Optimization Plan for Chronic Wasting Disease. You can check it out with the QR code. But basically, this is a tool for biologists to go in and look at their own state in relation to other states. So it keeps proprietary data safe, but it allows you to see, metadata for other states. And you can input things like the deer density in certain areas, where taxidermists are located, where captive deer herds are located, which are risk factors for the introduction of CWD. And you can model these things to see where you should go out and do surveillance. For the most likely place you're going to find CWD, if it comes to your state, you can focus your surveillance efforts in those areas. So this is just like a really cool tool that I like to show people. And we're going to stay with white-tailed deer, but switch gears a little bit and talk about COVID. So did anybody know that deer got COVID? Yeah. So we started testing. Our virology lab at the vet school was doing a SARS-CoV-2 project, and because we were receiving lymph nodes for chronic wasting disease testing, it was their idea. They were like, can we have your lymph nodes when you're done? And we can also test for COVID. And in doing so, they actually found that in a spike in SARS-CoV-2 infections in white-tailed deer in 2021, over 20% of the samples tested were positive for SARS-CoV-2. It trickled off after 2022. When that first happened, everybody was like, oh boy, deer could be a reservoir for this disease. It could spill back from deer to people or to other wildlife. But in 2022, it was back down to like 0.04% or 0.4%. So they're continuing to do this project to look at white-tailed deer, but it looks like they're not the reservoir we initially were afraid of, but it is interesting that they can be susceptible to the disease. And this is just a really cool, I think, online thing you guys can check out. I am sharing this PowerPoint, so all these QR codes that are flying by, you can look at them later too. But you can zoom in on all these different places around the world and see what animals are most affected in different areas. The green is a mink, deer obviously, is of course biased by what's getting sampled and tested in different areas, but I think it's really, really interesting to zoom in, and it's got a lot of good information about SARS-CoV-2 in wildlife on this website. So, the next question is always like, how did deer get COVID again? So we don't know, but we think it's probably through things like this, wildlife rehabilitation, where humans are working with animals, with wildlife directly, potentially captive cervid herds. They're managed by people. It most likely went from people to deer, but it's also possible that other indirect routes or even other wildlife passed it to deer. So the last thing that I'm going to talk about today is bobcats. I don't only do dead things. This is a live animal project that we're working on right now. So I'm the veterinarian for this project, and then there's Dr. Angela Fuller is USGS Extension Associate based at Cornell, and Josh is her postdoc, and Haley is the master's student on this project. So their goal is to look at density and movement of bobcats in New York State. The last study on bobcats, like comprehensive study, was in the 70s. So it's been a long time, like 50 years, since anyone's looked at bobcats in New York. So we want to know, how are the populations doing? And I said, I was happy to help, obviously. Very cool project. So I'm, running the sedation on the bobcats, and it's like, if we're going to have an animal in our hands under sedation, we should take blood, we should take feces, hair, ectoparasites, as many samples as possible. So I added this whole health aspect to the project, and it's still ongoing. So I don't have any data to show you, just a couple of cool pictures of bobcats. But these are animals that we've collared so far. So we have 17 animals, mostly in the southern tier. So Region 4, these are the regions that the DEC follows. So Region 4 has had quite a few. We've had 7 animals in that region, and then 7, 8, and 9, sort of Western New York. Regions 1, 2, 3, and 5 are not enrolled in the study. We didn't expect to find a lot of bobcats there. And then Region 6 is, but we haven't caught any yet. The study runs until the middle of March. But basically, if anyone's not familiar, we're collaring the animals with a GPS collar so that we can look at where they go and how long they spend in different areas. So this animal, when it was first sedated, it was sedated right here in this region, and this is a blow up of that. It spent a good amount of time the first couple of days after in that same region. But this is a 3-week snapshot of what the animal was doing, and as the points get more and more red, like all the way out here, that was the animal dispersing. Even, so one of the goals is to look at home range. We don't know the home range of bobcats. They estimated it in the 70s, but now we have much more advanced techniques that we can use to look at that. So we're getting there, which is really cool. And then the health, this is just the full suite of what we're looking at. So hemograms and chemistry is just sort of general health of the bobcats, and then a lot of infectious disease screening. The only ones that are done so far are the Cytoxoanopheles, and we haven't found it in any of the animals, which is good news. All of them are blood type A so far, which is, I don't know, interesting. Cats can be A, B, or AB, and they're all A so far, which is interesting. And then we're doing fecal floats, so looking at their parasites in their poop, and looking at their tick identification. One had some fleas, most of them have some ticks, so we're doing parasite ID with those animals. So I'm gonna sort of conclude with that. And so this is our website with the Cornell Wildlife Health Lab. You can use the QR code again. It's got a lot of great resources. It's got, all the faculty contact information too. Feel free to reach out to any of us. Definitely follow us on social media. We're pretty active with Twitter and Instagram, and then those are our fact sheets again in the middle. And that's all I've got for you. And that's my contact information, so feel free to reach out to me. And I'm happy to answer questions. Yes?
Did you see the area that the chronic wasting disease was in? Did you see that kind of expand in any correlation to temperature changes?
Oh, that's a good question. Not that I know of. Unfortunately, chronic wasting disease, there's some testing bias that sort of interrelates with temperature. So we always test during hunting season because that's when you can test other animals throughout the year, as they come in, like roadkills and things like that. But the vast majority of testing is done during hunting season, so you're only getting one temperature season. So it's very hard to tease out that data. Yeah, it's a good question, though. Yes, Marion.
With your bobcat study, you aren't doing any genetic testing to figure out whether you've got strong family groups or whether you've got dispersal genetic variability.
I should start like a GoFundMe. I collected hair. I'm collecting hair and blood from all of the animals, but we don't have any money for it right now. So I hope that we can do that in the future, but not right now. Yeah, it's a really interesting question. Yes, yeah, it's it definitely takes, yes. So we work with trappers across the state, and we're paying them to, rather than keep the animal for, the fur industry is still pretty strong in New York, so we're paying them rather than to keep the animal as fur to let us put a collar on it and keep it alive. So they check their traps, they call us when they have an animal, and we deploy as soon as we can, as soon as we get a call. They're required to check the traps at least once a day. And we generally need 3 people minimum to work on the cat. 4 to 5 is ideal because you've got, two people trying to put the collar on, put an ear tag on the animal. It's like a colored tag so that if it passes by a camera, we can get that kind of data too, like a trail camera. And then my sole job is like monitor the animal, make sure it does well under anesthesia, monitoring heart rate, temperature, and respiratory rate primarily, and then getting the biological samples. It's nice to have another person, but sometimes I do all of it. And then we always have a data taker too, because we're monitoring the heart rate and everything at certain intervals, at least every 5 minutes, and marking, what color tag did it get, and we're taking all, lots of measurements like the full body length, the tail length, the zygomatic arch length, and so a lot of morphometric measurements that they're keeping track of. So they have a data sheet, and it's really helpful to have a data taker. But it takes a little bit of a army. Yeah.
With the avian influenza one and marine mammals, did you see any change in how the disease progressed or its genetics between the avian and the
Yeah, so avian influenza is really well known for its ability to mutate and adapt to different hosts, so it's like changing all the time. And that's one of the sort of scares with it adapting to mammals, is that it could, make that ultimate leap to where it's easier to adapt to humans as well. So yeah, I don't personally do that research, but there's a lot of people looking at the genetics and how it mutates with every different species.
Just to follow up, is there any correlation between ocean temperature, one and that?
Yeah, so avian influenza virus can be pretty stable in the environment, but it's an RNA virus, so not as stable as DNA. And so, yeah, with warmer temperatures, it's less likely to survive. It survives better at cooler temperatures, so the warmer it is, the less length of time it's going to be infectious in the environment. So there's that. Yes.
Well, Lyme disease, have you been testing with all of these collections? Are you testing, and how does that affect the wildlife?
No, that's a great question. We aren't with the Bobcat Project. We probably could with like the ticks and things too. There's a group I know with the Department of Health and some of the domestic animal species, although this is a great example of how interdisciplinary work is super important, because there's people on the human health side looking for Lyme disease, people with pets like veterinarians screening for Lyme disease in dogs, but they don't talk to each other a lot. So those kind of things are always evolving, and I think we're doing better and better as time goes on. But Lyme disease is a good example where we were talking about looking at Lyme recently, and it was we still might in the future, but the lift was pretty heavy because the information's not readily available. There's no, like, when a veterinarian finds a Lyme disease case, it's not a reportable disease, so they're not required to put that in a database somewhere. So it's like, to learn the prevalence of Lyme disease in a certain area, you'd have to call every veterinarian and ask for all their records, and so it's kind of some diseases get the information's not easy to find. Yeah. And you had a question too?
How, like, with the bald eagles and like the lead, like, have you seen like backlash from like the hunting community saying like lead, like, or has
Yeah.
like did the Department of Conservation reached out saying like, hey, could you not?
Yeah, no, that's a great question. On that slide, so I think you'll have access, right, to this PowerPoint. On the slide with the lead, there's a QR code that goes to a website that has a video that was a partnership that we did with DEC and the Wildlife Health Lab, and it talks about non-lead ammo, and it is, yeah, it's definitely an issue. It's really hard to find. Like, you can use copper ammunition, but it's very difficult to find. It's more expensive. There's a supply and demand issue, really. So we can encourage it, of course, and practice what you preach type of thing. But yeah, there's a lot of people who either complain because it's too hard to get, or think it's not true. So it's hard, but we try to educate those people. And the video, I think, is a really good one because we worked with a member of the public who was like, this is a thing. I support it. It's hard to find copper ammo, but I do my best. I can't always, but and we're not saying you can't use lead ammo either. Maybe it's best not to, but if you clean up your gut pile and don't leave it in the environment and dispose of it appropriately, then you also reduce the risk to secondary scavengers. Yeah.
So, maybe a bit wild, but if you had like a really, if you magnetized it,
Okay. Yeah.
if you somehow fire it, you got a really strong magnet, would that take out the lead?
Lead is extremely soft metal and it doesn't even show up in the metal detectors in sawmills, so, yeah, no.
Cool question. Yes.
Are you working on making lead-free ammunition, making leaded ammunition illegal?
No.
California has put a ban on
Yes.
lead ammunition to certain areas because of the California condor ammunition.
Yeah, I don't foresee it in New York's immediate future. I think it'd be great if we could go that way eventually, but no, they're trying to incentivize non-lead ammo, like vouchers for if you buy copper ammo. But yeah, California, they're pretty progressive. They've outlawed lead ammo and lead fishing gear. So that's another major issue that the ammo gets all the backlash, but lead sinkers and things for fishing are also a big issue.
I think maybe education in terms of when you, as a hunter, shoot something, those lead fragments go into the food you're eating too, and
Yeah.
you're chopping down lead, and it's not good.
Feeding it to your children with developing brains.
Yeah, yep.
Well, I know in waterfowl, like, I'm certified to take people on refuges and stuff like that, and you're not allowed to use lead at all,
Yes, yeah,
so
yeah.
it is a restriction.
Well, thank you, Jane. This is
Yeah, you're welcome. Thank you.